Hello, and thank you for attending WPC Virtual and streaming my talk. My name is Mike Henderson, and I lead the Henderson Lab at Van Andel Institute. My lab investigates Parkinson's disease in cell and animal models in human tissue. We are a neuropathology lab, and we try to understand how normal brain proteins misfold and impact cell and network functions. And we evaluate potential therapeutics for their ability to modify pathology and neurodegeneration. Today, I'll be talking about tau co-pathology in Parkinson's disease. When we think about pathology in Parkinson's disease, we think about Lewy bodies and the misfolded protein that accumulates in these intraneuronal inclusions, alpha-synuclein. If we take a piece of human brain and stain it for misfolded alpha-synuclein, we can visualize this pathology. In this image, you can see two cortical Lewy bodies and many Lewy neurites labeled for misfolded alpha-synuclein. The presence of Lewy bodies is how neuropathologists know that a patient had Parkinson's disease dementia, or dementia with Lewy bodies. When alpha-synuclein aggregates into Lewy bodies in the substantia nigra, it leads to death of dopaminergic neurons there and subsequent motor dysfunction. However, Lewy bodies can be found in other regions of the brain, and alpha-synuclein aggregation may even begin in the periphery. As disease progresses, Lewy bodies can be found in cortical regions as well, as shown in this image. But I'm not here today to talk about alpha-synuclein. I'm here to discuss a pathology commonly associated with Alzheimer's disease, tau tangles. In addition to their association with Alzheimer's disease, tau pathology can be found in around 90% of patients with Parkinson's disease or Parkinson's disease dementia, as you can see by the staging here, with gray noting patients who had no pathology. An appearance of tau pathology in PD or PDD is associated with increased Lewy body pathology in the same regions and a worse prognosis for patients. In this image of human brain tissue, you can see the tau tangle and neuropil full of tau neurites after staining for pathological tau. This is actually the same piece of tissue that we had stained for alpha-synuclein pathology. So if you double label the same brain section for Lewy bodies and tau tangles, you can often find them in the same regions. And this is what we mean by co-pathology. Neuropathologists have staged Lewy body pathology through the disease process, and they found that alpha-synuclein pathology seems to begin in the hindbrain and olfactory bulb, but then it appears to spread in a stereotyped fashion up through the brainstem, eventually affecting dopaminergic neurons in the midbrain. After that, pathology spreads out through cortical regions over time. What about tau pathology? Well, it begins in the midbrain, affects memory centers in the temporal lobe, and then spreads through cortical regions over time as well. If we overlay the spread patterns of these two pathologies, you can appreciate the purple regions that are affected by both pathologies. Other than the early brainstem predominant phase of Parkinson's disease, you can see the spread pattern of these two pathologies is remarkably well conserved. This suggests that there are conserved mechanisms governing the spread of alpha-synuclein in tau pathologies, and that by studying these spread patterns, we may gain insight into the progression of disease. I've previously mapped out alpha-synuclein pathology in the mouse brain and found that pathology spreads through neuronal connections, but the gene expression, such as the expression of alpha-synuclein itself, controls how that pathology spreads. So we aim to extend that analysis to tau. We do this using a combination of quantitative pathology and network neuroscience in collaboration with our partners Danny Bassett and Eli Kornblath at the University of Pennsylvania. We conceived pathology in non-transgenic mice with a small amount of tau, misfolded, derived from human brain tissue. We inject this tau into the hippocampus and overlaying cortex and allow the mice to age. This initiates pathology in mice that's progressive and follows a defined spatiotemporal pattern through the brain. Eli and Danny then reduce the brain to a model of nodes and edges representing regions of the brain and axons which connect one region of the brain to another. They can then test mathematical models using graph theory to explain quantitative pathology patterns. Results of this project are shown here. Following those injections with misfolded tau, we quantify tau pathology throughout the brain at 1, 3, 6, or 9 months after injection. Blue denotes regions with minimal pathology, and red denotes brains regions with extensive pathology. There's very little pathology at 1 month after injection, but pathology increases by 3 months following injection near the injection sites and begins spreading to the opposite side of the brain at 6 months post-injection. By 9 months, pathology can be found throughout many regions of the brain. Since all of this is quantitative data, we can plot it out as shown here, with each dot representing a region of the brain. Eli can then seed pathology into the same nodes of his computational model and estimate how much pathology should spread along those edges to other regions of the brain. When he does that, we get predicted pathology for all the same regions, and we plot those values against actual pathology. We find that we're able to predict to a substantial degree how much pathology is in each region over time. Eli has found that the best model incorporates spread along neuronal connections in both the forward and backward direction, with a preference for backward or retrograde spread in cells. However, some regions still get more pathology than we would expect based on anatomical connectivity alone, and some get less. And we can think of these differences as a measure of vulnerability. When we take those vulnerability values and average them over time and across brain hemisphere, we get relative vulnerability values as shown here, with more vulnerable regions shown in red and less vulnerable or resilient regions shown in blue. We can then compare those vulnerability values to regional gene expression and pull out gene expression patterns that are associated with vulnerability or patterns that are associated with resilience to tau pathology in the mouse brain. We hope that these data can be used to understand vulnerability and resilience to pathology in the brain. In summary, I hope I've conveyed to you that tau is an important co-pathology in Parkinson's disease, correlating with progression to dementia, and that it's valuable to study tau pathology in the context of Parkinson's disease. Tau spread can be modeled in mice through quantitative pathology and network neuroscience. We find that tau spreads from region to region along neuroanatomical connections, but is modulated by vulnerability factors, which could include things like neurons or glia type, or certainly the type of connections. And you'll hear more shortly from Shane Littlelow about the role of glia in neurodegeneration. But these studies were in mice. Can this help us understand human disease? Recent imaging studies in human brains have found that atrophy or pathology patterns in neurodegenerative diseases can be explained by using similar mathematical models based on spread through human brain anatomical connections. I've noted several examples here for Parkinson's disease and for tauopathies, including Alzheimer's disease and frontotemporal dementia. It's our hope that by understanding this spread, we can better estimate disease prognosis and evaluate the impact of therapeutics on disease progression. I want to close by thanking you for your attention and thank those who contributed to this study, including my postdoctoral mentors, Virginia Lee and John Trojanowski, and our computational partners, Danny Bassett and Eli Kornblath. I also want to thank the patients who donated the tissue to further research into this disease. This study was funded in part by the Michael J. Fox Foundation. And if you're interested in learning more about our ongoing research, please visit our lab website or follow us on social media. Thank you.